DNA contains instructions to make proteins. That means DNA contains genes. A gene is a complete string of nucleotides containing a region that codes for an RNA molecule. This region begins with the promoter and ends with the terminator regions. Genes also contain regulatory sequences that can be found near the promoter or at more distant region. During initiation, the promoter region of the gene functions as a recognition site for the enzyme RNA polymerase to bind. This is where majority of the gene expression is controlled. Binding causes the DNA double helix to open. Then, during elongation, the RNA polymerase slides along the template DNA strand as a complementary bases pairs up. The RNA polymerase links the nucleotide to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the termination region of the gene, then the messenger RNA synthesis is complete. Now the RNA polymerase template DNA strand and the newly synthesized messenger RNA dissociate from each other. The RNA which is synthesized during transcription include regions called exons that codes for proteins and non-coding regions called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed. This is undergone by modifications such as 5' capping and 3' poly A tailing. This process is called intron splicing. And the splicing mechanism is carried out by a complex of proteins and RNA together called spliceosome. This complex removes the introns and joins the exon segments. Now the RNA leaves the nucleus through nuclear pores and enters into cytoplasm for translation.